Hello friends, welcome back to my YouTube channel Nasrin Padasali. So today we are going to discuss the previous year question asked from Bioinformatics. So this will be really helpful for those students who are preparing for GATE Biotechnology. So from 2006 till to 2021 year, I just gather the question asked only from Bioinformatics because Bioinformatics is of course an important topic, but many of you will be skipping this particular portion in your GATE BT syllabus. So in this video we are going to discuss the bioinformatics question asked in this many years. First question, calculate the percentage sequence identity for a pairwise alignment given below. So this is what I think they had given. So the solution is that they had given two sequences. One is hello and another sequence is yellow. And the formula to identify the person identity between two sequences would be, so first you need to know number of identical position. That is here 4 positions are identical between the word hello and yellow, which in case the word the yellow ELLO is present in sequence number 1, which is hello, and the word ELLE is also present in the word yellow. Okay, and next divided by total number of position, and of course in hello total number of position is 5 HELLO, whereas if you take the word yellow the total number of position is 6. So you need to take the total number of position is 6 multiplied by 100. So total number of identical position is 4 and total number of position is 6. So you need to divide 4 by 6 into 100, then you will be getting a percentage of sequence similarity, that is sequence identity between this particular sequence. So if the question has been asked, you need to know this particular formula. Next question is, they ask us to find the number of unrooted tree in a phylogeny of 5 sequence. So first you should know whether they are asking the number of rooted tree or unrooted tree, and next thing how many sequence. Okay. So this is a simple thing, so you can remember this particular thing. Anyway, I am going to teach you a formula also. So this is a number of sequence, based on number of sequence, a possible number of rooted trees and unrooted trees. Since in this question they are asked the number of sequences 5 and they are asking unrooted trees, so the correct option answer is 50. So let me teach you formula, so this will be really helpful. So in order to find the total number of rooted trees, the formula is 2n, n in the sense number of sequence minus 3 the whole factorial divided by 2 to the power of n minus 2 multiplied by n minus 2 factorial. Okay. Then if they ask us to find the number of unrooted trees, the formula is simple. 2n minus 5 the whole factorial divided by 2 to the power of n minus 3 multiplied n minus 3 factorial. So since in this particular thing they are asking us to find the unrooted trees and the number of sequences 5. So you need to simply put the 5, then 10 minus 5, 5 factorial, then divided by 2 to the power of 2 multiplied by 2 factorial. Okay. So 2 to the power of 2 is 4 into 2 factorial. So you should not take it as a 8 factorial. You need to take factorial only for 2. Therefore the answer is 50. So 5 factorial means 5 into 4 into 3 into 2 into 1 and 4 multiplied by 2 factorial 2 into 1, then the correct option is, the answer is 50. Next question, which one of the following tool is used to compare all possible 6 open reading frame of a given nucleotide query sequence? Query sequence means the sequence which you have in your hand is called as query sequence with all available 6 open reading frame of nucleotide sequence in the database, like that they are asking. The correct answer is what? Okay, so that's what they are asking. So here you should know what is meant by BLAST. The full form of BLAST is Basic Local Alignment Search Tool. So this particular software finds a region of local similarity between the sequences. So it is a bioinformatics program which generally compares the nucleotide or protein sequences to the sequences which was already present in the database. Database is nothing but it is a hub where, like you, many of you will be submitting the sequence. Okay, so the sequence which you have in your hand is called as query sequence, and the sequence which is present in the database is called as target sequence. And this BLAST program, then after comparing, it will be calculating a statistical significance of matches existing between two sequences. So BLAST is used to infer the functional and evolutionary relationship present between the sequence. At the same time, BLAST is also helpful in identifying the member of gene families. Okay, so this is what I am telling you. So this is a sequence which you are having in hand and you will be comparing in the sequence present in the database. So these are the list of BLAST programs. So first is BLAST P. BLAST P is a software program which compares an amino acid query sequence against a protein sequence database. BLAST N, N for nucleotide, which will be comparing a nucleotide query sequence against a nucleotide sequence database. Whereas if you take BLAST X, it will be comparing a nucleotide query sequence which will be translated in all reading frame. So the message in DNA will be passed into mRNA, and from mRNA the code will be transferred to amino acid code. The process is called as translation. Okay. Again a protein sequence database. Okay. So BLASTX program can be used in order to find a potential translation product of an unknown nucleotide sequence. If you have a DNA sequence, that is a nucleotide sequence in a hand, if you want to find what is the potential translation product, means that at that time you can use BLASTX. Next program is TBLASTN, which will be comparing a protein query sequence against a nucleotide sequence database, which will be translated, not a simple nucleotide database, it will be translating into all reading frame, that is the nucleotide code will be transferred into an amino acid code. Next program is TBLASTX, which is a combination of BLASTX and TBLASTN. So this TBLASTX will be comparing the 6' translation of nucleotide query sequence against a 6' translation of nucleotide sequence database. So both your query sequence as well as the sequence present in the database are being undergoing the process called as translation. Okay, so the correct answer for this question is TBLASTX, and there are three other important BLAST programs. So first is MEGABLAST, which is a program optimizing for analyzing a longer sequence. If the sequence length is very long, then you can use MEGABLAST. At the same time, you can use only for DNA sequences. You can't use other than for mRNA sequences. And this MEGABLAST can be used if the sequences are closely related. Okay, next is PSI-BLAST. So PSI means position specific iterated blast, and this particular BLAST program is generally used for protein similarity searches. Next is PHI-BLAST. PHI means pattern hit initiated blast. So this particular BLAST program is generally used if you want to find any pattern or motif in your protein. So this is a simple thing I had given, friends. So only BLAST in, that's a comparison between nucleotide and nucleotide, whereas for the rest of the BLAST program the comparison is made between protein and protein. If you take TBLAST in, the query type will be protein, which is an amino acid sequence, whereas in database it is a nucleotide. Okay, and then the database is translated into protein. So here the nucleotide will be translated into protein, that is a 6' translation. Next, in BLASTX program, the query type will be nucleotide. Again, the queries are translated into protein, which is nothing but the DNA code will be translated into amino acid code, whereas the database will be a protein sequence database. And in TBLASTN, both the query type as well as the database, the sequences are translated into amino acid sequences. Okay. Next, like BLASTA, the other program is called as FASTA. So FASTA is a DNA and a protein sequence alignment software. Okay, so these are the list of FASTA programs. So first is FASTA, which is comparing a protein sequence to another protein sequence present in the database, and also it compares a nucleotide sequence to another nucleotide sequence present in the database. Next FASTA program is FASTX and FASTY. It performs a search by comparing a nucleotide sequences to a protein sequence database. So the query is nucleotide sequence. It will be comparing the query to a database of amino acid sequence. Okay. Next is search. It performs a Smith-Waterman alignment, which is nothing but it's a kind of alignment. So between a protein sequence and another protein sequence, or else between a nucleotide sequence and another nucleotide sequence. So search perform a local alignment. So in the upcoming slides, I'll be definitely explaining what is the difference between local alignment and global alignment. Next is search, which will be comparing a protein sequence or a DNA sequence to a sequence database using a global alignment. So search is using a local alignment, and search of FASTA program is by global alignment. Next is called GL search. So same, it will also compare a protein or DNA sequence to a sequence present in the database, but here the alignment are global in query and local in the database. So both the global and local alignment are possible with regarding to GL search. That's why they are giving G and L, global in query and local in database. So this is the difference between local alignment and global alignment. Local alignment align only a part of sequence, whereas global alignment align entire sequence, which is called as end-to-end alignment. Local alignment can be used for dissimilar sequence or partially similar sequence or sequences with different length. Okay, so those sequences that are suspected to contain a region of similarity, you can use local alignment, whereas global alignment is generally used for similar sequence. At the same time, those similar sequences will have to be of same length. Only then you can approach global alignment. Okay. Next, local alignment is generally used for divergent sequence or distantly related sequence, whereas global alignment is used for convergent sequence or closely related sequence. Next, local alignment is used for finding either conserved pattern in DNA sequence or conserved domain or motif in case of protein. Whereas global alignment is usually done for homologous gene comparison of two genes with same function. The example of local alignment is BLAST, L-align, emboss water, whereas example of global alignment is emboss needle. Next, this is what the local alignment. It will be aligning only few portion of the sequences, whereas global alignment it is starting from 5' to 3', that is end-to-end alignment. Next, identify the file format given below. So in bioinformatics, there are different file formats since there are different database being available. So first is NBRF or PAF format. So the sequence that are in this particular format begin with a two-line description. Okay, there is some kind of description regarding to sequence like which organism the DNA was isolated from, like that, followed by a line of sequence data. So the description line is distinguished from the sequence data. So sequence data here TYPL is a sequence data, and the description line is something that is related to size of DNA and from the which date sequence was uploaded in the software, like that. The description line is distinguished from the sequence data by starting with P1 greater than symbol and P1, and the second line contains comment, and also the sequence is terminated by asterisk symbol. Okay, so this is an example. Next format is IG format. So sequence file in IG format contains several sequence, not only one sequence, it may contain two or more sequence, and each sequence will be consisting of number of comment line. Okay, and each comment line will be beginning with a semicolon, and after this semicolon and comment line is followed by a line with a sequence name. So sequence name means it might be another name like EAB002. It's a code word. Okay, and this particular sequence name will not contain no spaces, and the sequence present in this particular IG format are terminating with a terminated character, 1 in case of linear DNA or 2 in case of circular DNA. Next is GenBank format. Sequence file in GenBank format also contains several sequences. So one sequence in GenBank format starts with a line containing the word locus. So here you can able to see I had given an example of GenBank format, and a number of annotation lines will be there. Annotation says whether it is a DNA or RNA. In case RNA, whether it is a mRNA or tRNA, what is the size, whether it is linear, circular, when it was published, and definition like from which organism the thing was identified, and also accession number, like that. And the start of sequence, that is a nucleotide sequence, is marked by a line containing the word origin, and the sequence is terminated by two slashes. Okay. Next is GCG format, and the sequence file in this particular format contain exactly only one sequence. If you take the sequence file in IG format or in GenBank format, it contain more than one sequence, whereas in GG format it contain exactly only one sequence. Okay, and it begins with the annotation line, and the start of sequence is marked by the line ending with two dots. So here you can able to see. Here you can able to see the start of sequence is marked by a line containing two dot. In the line also that is this two dot line also contain a sequence identifier, which is accession number, the length of the sequence, and a checksum. After giving that, then the line will be ending with a two dot, and after that the sequence will be fine. Next is FASTA format. The sequence in FASTA format begin with a single line description followed by a line of sequence data. The description line is called as def line, and it will be distinguished from the sequence data by a symbol called greater than symbol. And the text in FASTA file format should not exceed 80. It should be shorter than 80 character length. And next important thing is that blank lines are not even allowed in the middle of FASTA input. Next, this is EMBL format. So sequence file in EMBL format also contains several sequence. Uh, so only in GCG format exactly one sequence is present, whereas in another format like IG format, GenBank format, EML format, more sequence is present. So the sequence is entry. So one sequence entry starts with the identifier line called ID, followed by a further annotation line like mRNA or what is the size of sequence, and a start of sequence is marked by a line starting with SQ, and the end of sequence is marked by two slashes. Next, GDE flat file format. This is a minimal file format which will be retaining only the name and the sequence data. It is used by GDE in order to create a temporary file, and there is no direct way to save this particular format. The name in each sequence in GDE file format indicate this type of data. That is, hashtag indicate DNA or RNA, percentage indicate protein, at indicate mass sequence, whereas this particular apostrophe indicates text. Next, in bioinformatics, next question, in bioinformatics, the term BLAST refers to computational tool for sequence homology searches as well as for alignment. You can use BLAST. So, BLAST finds a region of similarity existing between the biologic sequence. This BLAST program generally compares a nucleotide sequence or a protein sequences to the sequences present in the database, and also it calculates statistical significance of matches. So these are the use of BLAST program. Like if you have any newly isolated protein, newly isolated amino acid sequence, you don't have any idea about what is the function or what is the name means, then by using this BLAST program, you can identify the name and function of protein or nucleotide, nucleic acid or DNA. At the same time, by using this BLAST program, you can perform pairwise alignment, and by using this BLAST program, you can design your PCR primer. At the same time, you can also check the specificity of your PCR primers. These are the application of BLAST, friends. First thing, it can be used for DNA mapping. So DNA mapping between a known sequence and unknown sequences, and also it is used for domain location. Like you can able to locate the domain and protein of interest. At the same time, it can be used for comparison. Like you can identify the similar gene present or a function, similar function present between species. And the next function is identification of species. Like you can identify a species by comparing the sequences of DNA between a different organism. The last, you can also establish the phylogeny tree. So this is the example with differentiation between BLAST and FASTA. So BLAST was given by this particular person, Miller, Warren, Eugene, David J. Lippmann at the National Institute of Health, like NIH, in 1990. And FASTA were given by David J. Lippmann and William R. Pearson in the year 1985. And BLAST generally follows a local alignment, and FASTA also follows a local alignment, but after next stage it will be extending to global alignment. So both the global alignment and local alignment are possible in FASTA. Next, the gap between a query sequence and target sequence are not even allowed in BLAST, whereas in FASTA it is allowed. BLAST is generally for highly similar sequence or closely related sequence, whereas FASTA is for less similar sequence or more distantly related sequence. Next, so we were speaking about the sequence alignment. So sequence alignment between two sequences is performed by three methods: dot matrix analysis, dynamic programming, and the last method is widely used, that is FASTA and BLAST bioinformatics programs also using this particular method, which is called as word size or K-tuple method. So word size is also called as K-tuple method. So this method aligns two sequence very quickly. First, by searching for an identical short stretch of sequence called as word. This is also called as K-tuple. So this is how the BLAST program will be. BLAST starts with finding a perfect sequence match of length by giving a word size, and these are default word size. So BLAST program generally go for overlapping words. So here you can able to see this is amino acid query sequence. So this is word 1 is PQG, word 2 is QGE, word 3 is GEF. Like that it will be keep on searching the entire sequence. And the nucleotide, for nucleotide search using megablast, the default search size is 28, like 28 nucleotide ones. So here it can able to see three nucleotide. This is a 3 nucleotide length, whereas this is for protein. So for amino acid sequence, the default word size is 3, whereas for nucleotide sequence by using BLAST 10, the word size is 11. 11 nucleotides it will be searching, whereas for nucleotide sequence database using megablast, the minimum word size or word size is 28. Whereas FASTA, the default word size is given, but FASTA generally go for non-overlapping word. So say this is a nucleotide query sequence, means here the first word will be ACG and the next word will be TAC, like that. It won't go for ACG and CGT and GTA, like that it will not go. And the nucleotide default word set for nucleotide sequence is 3 to 6 nucleotide in length, whereas for amino acid sequence it is 1 to 2 amino acid in length. Next question is, accession number is a unique identifier number assigned to. So accession number is a unique identifier number which is assigned to single database entry for both for DNA, protein, RNA. So accession number in bioinformatics is a unique identifier given to a DNA sequence or a protein sequence record. It is generally a combination of letters or number, like single letter followed by 5 digit or 2 letter followed by 6 digit, and that accession number do not change. So this is a format of GenBank accession number. So there are two type of things, friends: GenBank and Refseq. So GenBank is not a curated one, whereas Refseq is a curated one. And in GenBank, the sequences will be submitted by the author, whereas in Refseq, NCBI will be creating that from the GenBank. Okay, and with regarding to GenBank, there is no limit to species. All the DNA isolated from different species can be submitted. The nucleotide sequence can be submitted in GenBank. Whereas in reference sequence, you can find the nucleotide sequence or amino acid sequence or RNA with regarding only to model organism. And GenBank is redundant in nature, whereas reference sequence is not redundant in nature. And next is regarding to accession number. GenBank accession number never include any underscore, whereas ref sequence includes accession number present in the accession number of nucleotide sequence or protein sequence present in ref sequence database includes underscore. So this is for nucleotide. The format of GenBank accession number is 1 letter followed by 5 numeral, or 2 letter followed by 6 numeral, or 2 letter followed by 8 numeral. Whereas in protein, the format of protein sequence in GenBank accession is 3 letter followed by 5 numeral, or 3 letter followed by 7 numeral. So this is a format of ref sequence accession number. So ref sequence accession number do not follow the naming convention as set by INSTC, which is nothing but is a international union which includes your GenBank, DDGB, like that. It will contain numerous database. So the ref sequence accession number consists of 2-letter prefix followed by a underscore. And the ref sequence record are generally classified into two types. One is known ref sequence, which is manually reviewed by the staffs working in NCB or collaborator, whereas in model ref sequence, it will records are produced in this model ref sequence by using automatic pipeline. This accession prefix for reference sequence. NC underscore means complete genomic molecule. NG underscore mean incomplete genomic molecule. NM underscore mean mRNA. NR underscore mean non-coding RNA. NP underscore mean protein. NT underscore and NW underscore mean genomic contic or scaffold. Okay, so if a sequences from scaffold sequences submitting means in a reference sequences, then the sequence will be starting with NW or NT followed by an underscore. So this is for known reference sequences, whereas for model reference sequences, it's generally starting with XM for mRNA, XR for non-coding RNA, and XP for protein. So the next question is an example of program constructing a phylogenetic tree. So they had given the correct answer is phylip, friends. So these are the general purpose packages or software analyzing for constructing phylogenetic tree. So phylip, Mega, Phylowin, next is Embos, ETE, CView, Cruxor. Software is used for constructing phylogenetic tree. Whereas if you take FRAC, this is used for assembling shotgun DNA sequence data. PRODOM is used for a database for identifying protein domain family, and this PhD-seq is used for prediction of secondary structure. Then a suitable substitution matrix to align a closely related sequences. So they are asking which substitution matrix will be used for aligning closely related sequence. The correct option is PAM40 and BLOSUM 80. So substitution matrix are matrix which is generally used to score aligned position in a sequence of alignment procedure. So substitution matrices are generally used in BLAST program, and there are two common methods of the substitution matrix. First method is PAM, person accepted mutation, or percent accepted mutation or point accepted mutation, which was given by Margaret Dayhoff, whereas BLOSUM, which is a block substitution matrix, which is given by Stephen and Hennicock. Okay, and this is a differentiation between PAM and BLOSUM. PAM matrix are used to score alignment between closely related protein sequences, whereas BLOSUM are used to score alignment between evolutionary divergent protein sequence or distant protein sequences. Next, PAM is based on global alignment, that is N2 alignment, whereas BLOSUM is based on local alignment. Next, high number in PAM matrix naming denotes there is a greater evolutionary distance. Okay. See, why I don't select PAM 250 means here they are asking closely related sequence. That's why I have given PAM 40. If you take PAM 50, we can't able to use PAM 250 for closely related sequence. We can use PAM 250 for distantly related protein. That's why high number of PAM matrix naming denotes a greater evolutionary distance. That is, PAM 250 is used for more distant sequences when compared with PAM 120. So here BLOSUM means high number in BLOSUM matrix naming denotes that is the high sequence similarity and a small evolutionary distance. So both are having an opposite meaning, friends. So if you take BLOSUM 80 is used for closely related sequences, then BLOSUM 62.
So here you can able to see point accepted mutation is used for species having a deletion or insertion at any particular point in a nucleotide sequences, and block substitution matrix is used for species whose sequences are undergone any kind of substitution. And if you take PAM40 means 40% sequence are mutated and remaining 60% are conserved in nature. So that's why we are using this PAM40 matrix in case of closely related sequences. Whereas if you take BLOSUM 80% sequence are conserved and rest of 20% are mutated in nature. Next, PAM40 and BLOSUM 80 are generally used for less divergent or closely related species for performing alignment. Whereas if you take PAM250 or if you take BLOSUM 40 et al., you can use these two matrices in case if you want to align distantly related sequence or sequence which don't possess any similarity. And next question is that in an affinity gap penalty model, if the gap opening penalty is -20, gap extension penalty is -4 and gap length is 8, the gap score is. So you should know gap penalty is classified to 5 types. So first is constant, next is linear, next is affine, next is convex, next is perfect. First is constant. So with regarding to constant gap penalty model, a fixed negative score is given to every gap regardless of its length. So if you take this particular sequences, here there are 3 gaps. Whatever the length of gap will be, whether there will be a gap in 3 nucleotide, whether the gaps are gap in a 10 nucleotide, each match will be given a score of 1. Okay, so that is total number of sequences is given. So here totally 7 nucleotide are matching. So 7 minus 1. So gap. So totally 3 gaps are there. So that's why they had given 1. So okay, 7 minus 1 you will be getting 6. Whereas if you take linear will be taking into account of length of each insertion gap or deletion gap. So here if you take same thing, so there are 7 nucleotide matching, there are 3 gaps, so you need to put number 3 because for each gap you need to give a score of 1. So 7 minus 3 you will be getting 4. And affine model you are going to see gap opening penalty will be there, gap extension penalty will be there, and this is a formula to find the gap score in case of affinity gap model, that is G is equal to GI plus K plus GE. So G is a gap score to be calculated, G is a gap insertion penalty which is -20, K is a gap length, number of nucleotide missing, and G is a gap extension penalty that is -4. So if you apply over here, then you can easily able to calculate what I mean to say. Okay. Next is with regarding to last gap penalty type is profile. So it is a powerful tool for detecting the protein homology relationship. So generally Clustal W MAFET adopt this kind of gap penalty determination for doing their multiple sequence alignment. So how many rooted and unrooted phylogenetic trees are respectively possible with 4 different sequences. Already we dealt with the formula. So you can use the formula, just calculate and tell me in the comment section whether that is you had calculated that particular thing is correct or not. Next is percent of similarity and identity respectively between 2 peptide sequences given below. They are asking us to find out what is the percentage of similarity and the identity. So they have given 2 peptide. So if you take in this particular case, total number of amino acid is 15, and among this 15 amino acid, 9 amino acid are identical. Say in the peptide 1 alanine is present is number 1, and here peptide 2 also is alanine present. And if you take over methionine is present in peptide number 30, peptide 30, peptide 1, 13th position methionine is present, and also in peptide number 2 methionine is present in 13th position. So 9 out of 15 amino acids are identical. So simply you can calculate percentage of identity. It is 9 by 15 into 100. So total percentage of identity is 60. Next, in peptide 1 and peptide 2, if you take arginine in peptide 1 and glutamine in peptide 2 are identical. Next, they are similar amino acids. So what is mean by similar amino acid means they will be having a similarity in their structure. So that's why they are called a similar structure. So same if you take leucine in peptide 1 and isoleucine in peptide 2, both are similar. And at last, if you take glutamine in, that is glutamate in peptide number 1 and asparagine in peptide number 2, both are similar. So among these 3 amino acids are similar in nature. So similar amino acid means they will be having the same molecular form, but they will be differing in their structure. Okay. So you should know arginine and glutamine, leucine and isoleucine, glutamine and asparagine are similar amino acids. So totally 3 amino acids are similar. So therefore 9 plus 3 divided by 15 into 100. So 9 saying 9 amino acids are identical, 3 amino acids are similar. So this is how you need to find the percent of similarity between 2 amino acids. Next, so this video is little bit lengthy, so we'll be discussing the rest of question in the part 2. So please follow that particular video also. Thank you friends, thanks for watching this video.